Welcome to Mr. Chalk's Revision Tips.
When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just to take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make, and if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
Non-coding DNA corresponds to the various portions of the genomes that do not code for amino acids. So some non-coding DNA sequences are known as transcription factors, such as regulatory genes, while other areas of non-coding DNA we still don't know the function for. So the genome within eukaryotic cells, so that's cells with a nucleus, contain many non-coding sections of DNA. So like we said, that do not code for any amino acids. So non-coding DNA can be found between genes as non-coding multiple repeats. So this means that they contain the same base sequences repeated multiple times. Non-coding DNA can also be found within genes as introns. So the coding extrons can be separated out by one or more introns. So during transcription, eukaryotic cells transcribe the whole gene, so all of the introns and all of the extrons, to produce pre-messenger RNA or immature messenger RNA. So before pre-RNA can exit the nucleus, the non-coding section, so the introns, are removed, and the coding section, so the extrons, are joined together in a process called splicing. So non-coding DNA is not present in the mature messenger RNA. So the mature messenger RNA is kind of the crucial step within protein synthesis. So at this stage, the introns and any extrons that are not required have been removed from the messenger RNA by splicing, leaving only the extrons that code for the amino acids that make up a particular protein. So non-coding DNA is never translated into proteins, most importantly. So we'll just go and have a look at this question. So DNA codes for proteins within the cell. So some regions of DNA are described as non-coding. So explain, the important bit there is the explain part. Why some regions of DNA can be described as non-coding. So like we've said, that there's primary editing of those things that are going to be transcribed. So it's not present in the mature messenger RNA. So it's not translated and can cause things like regulatory factors. So non-coding regions of DNA show more variation than coding regions. This makes non-coding regions useful for DNA profiling. So suggest why those non-coding regions show more variation. Right, so it's only a suggestion, so we don't need to go into that much detail. So it's just something like the fact that in evolution or reproduction, they're not selected against because they do not affect survival. So therefore, it's not that crucial that they remain completely stable all the time. So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.